Okay, so welcome back to this next video in which we are discussing Cys-Loop Receptors. Okay, right, so we've now discussed the structure of Cys-Loop Receptors in detail, and we've discussed the gating mechanism for Cys-Loop Receptors. We've discussed how, when the ligand binds to Cys-Loop Receptors, what actually occurs to cause the opening of the channel. Okay, what we now want to turn our attention to is a more in-detail discussion of the different types of cis-loop ligand-gated ion channel. Okay, so we're now going to go into more detail looking at those 4 families of cis-loop ligand-gated ion channels found in humans and other mammals, and we'll also look at the glutamate-gated chloride channel, which is found in worms in a little bit more detail, and how the drug Ivermectin, which is an agonist for that glutamate-gated chloride channel, can be used to treat certain parasitic worm infections. Okay, we won't be talking about ELIC and GLIC anymore than we did when we went through them briefly. Okay, right, so let's now start by talking about the nicotinic acetylcholine receptor family. Okay, so again, as I've been saying all the way through, this is not a single type of receptor. This is an entire great family of receptors. Okay, so it would be lovely and simple if there was just one nicotinic acetylcholine receptor subunit, and then to build a nicotinic acetylcholine receptor, all you had to do was build a homopentamer of this nicotinic acetylcholine receptor subunit. It's not the case. In reality, there are 16 different nicotinic acetylcholine receptor subunits in humans, okay? And these will all have the same basic structure of all cis-loop ligand-gated ion channel subunits, that structure that we have looked at now in a huge amount of detail. So that extracellular domain with those 2 sheets, okay, the outer sheet, which is the positive side, and the inner sheet, which is the negative side, the 4 transmembrane alpha helices, and then that intracellular loop between the 3rd and the 4th membrane spanning alpha helix. They will all have that same basic structure, but they will all differ in their primary sequence of amino acids, so they are fundamentally different proteins. Okay, so let me tell you about the naming of these 16 different subunits for nicotinic acetylcholine receptors. Okay, so we split them up into the alpha and beta families, which contain multiple different nicotinic acetylcholine receptor subunits, and then there is the gamma, delta, epsilon families, which only contain 1 subunit. Okay, so there are 9 different alpha subunits of nicotinic acetylcholine receptors in humans. Okay, but they're not called alpha 1 to alpha 9, unfortunately. There is one little break in this. Okay, they are called alpha 1, alpha 2, alpha 3, alpha 4, but it goes up to alpha 10, and the reason for that is that there is no alpha 8 in humans. Okay, there is an alpha 8 nicotinic acetylcholine receptor subunit, but it's not found in humans, it's found in birds. Okay, so as far as talking about the nicotinic acetylcholine receptor subunits in humans, you have alpha 1 all the way up to alpha 7, then alpha 9 and alpha 10, but there is no alpha 8. Okay, so those are the 9 alpha subunits of nicotinic acetylcholine receptors found in humans. And you also have some beta nicotinic acetylcholine receptor subunits. Again, there are 4 of these in humans, and they are simply called beta 1 all the way up to beta 4. Okay, that takes us up to now a total of 13 nicotinic acetylcholine receptor subunits, and then the final ones are just the gamma, delta, and epsilon, which are also found in humans. Okay. Right, so here are the names of the 16 different subunits that are all cis-loop ligand-gated ion channel subunits, which you can use to build nicotinic acetylcholine receptors, basically. Okay, so there are a huge great number of different nicotinic acetylcholine receptor pentamers that you could potentially build. Okay, because all you have to do is put 5 nicotinic acetylcholine receptor subunits together to build a full nicotinic acetylcholine receptor pentamer. So obviously there's a huge number of different ways of doing that. Okay, and not all of the possibilities are obviously actually found in life. So what I'm now going to just go over is some really important examples of nicotinic acetylcholine receptor pentamers that are actually found in humans, basically. Okay, so the constructed pentamer. So these are the 16 different individual subunits. Now what we're going to go over is the most important pentamers. Okay, so we've already done this neuromuscular junction one up here. Okay, so this is the first one and a really, really important one. Okay, so this is the one present in skeletal muscle cells and which responds to the acetylcholine released by alpha motor neurons. Right, and will open and allow sodium ions into the skeletal myocyte to depolarize the cell, and therefore making it more likely that the skeletal muscle cell will fire an action potential. Now let me talk about the ganglionic type of nicotinic acetylcholine receptor pentamer. Okay, so next up is the ganglionic type. Okay, so this is slightly easier than the neuromuscular junction nicotinic acetylcholine receptor, because this only contains two different types of nicotinic acetylcholine receptor subunit. Okay, it contains the alpha-3 type subunit, okay, and you have two of those, and then it also contains the beta-4 nicotinic acetylcholine receptor subunit, and you have 3 of those. Okay, so alpha-3 and beta-4, and I apologise for subscripting that 4. Okay, some people will subscript the numbers here, but I've been not subscripting them previously, so I've made a mistake there by subscripting that. Imagine that it's not subscripted. Okay, right, so you have 2 alpha-3 subunits and then 3 beta-4 subunits. You put these together and that is going to make the ganglionic type nicotinic acetylcholine receptor. Okay, now let me show you how they're going to be arranged relative to one another. Okay, so again I'm going to draw the nicotinic acetylcholine receptor as viewed from the extracellular side. Okay, so here are the 5 different subunits that are going to make up the nicotinic acetylcholine receptor pentamer. Okay, right, so basically the 2 alpha-3 subunits of the nicotinic acetylcholine receptor ganglionic type are going to be separated by a single beta-4 subunit. Okay, so here is one alpha-3 and here is the other, and you're going to have a single beta-4 subunit in between them, basically. Okay, and then these 2 spaces will be filled in with the remaining beta-4 subunits, like so. Okay, so I'll just colour this in. So we'll colour in the alpha-3 subunits of this ganglionic type nicotinic acetylcholine receptor in green here. So those are the alpha-3 types. Okay, and then we'll colour in the beta-4 ones in blue here. Okay, now all that remains for me to say is how many binding sites does this ganglionic type nicotinic acetylcholine receptor have, and where are the binding sites? Okay, so the answer as to how many it has is that it has 2, okay, and that they are at the positive sides of the alpha-3 subunits. Okay, so remember how the positive-negative works. The negative side of a subunit is always clockwise of the positive side. So if we do this for the alpha-3 subunit here, this is the positive side of this alpha-3 subunit, this is the negative side. So this is where you'll have that outer sheet, and this is where you'll have that inner sheet. For this alpha-3 one here, this is the positive side, and this is the negative side. Okay, and basically you're going to have one ligand binding site for acetylcholine here between the alpha-3 positive side and the beta-4 negative side, and another over here, again between the alpha-3 positive side and that beta-4 negative side. Okay, so again I'll stress the point that although there are interfaces here, here and here as well, they are not going to have the correct residues on those 6 important loops to actually be able to coordinate a molecule of acetylcholine, basically, and therefore they're not actually acetylcholine binding sites. So the alpha-3 to beta-4-3 Hetero Pentamer, or the ganglionic type nicotinic acetylcholine receptor, is only capable of binding 2 acetylcholines overall. Okay, so again, acetylcholine will be released by our preganglionic neuron onto our postganglionic neuron. It will bind to these ganglionic type nicotinic acetylcholine receptors. It will cause the nicotinic acetylcholine receptor to open in that mechanism that we have now discussed. All of the extracellular domains will rotate. It will be transferred to the M2 helices. The hydrophobic girdle will be broken, and that will then open the pore, and then it will conduct sodium ions and potassium ions, but principally, of course, sodium ions are now going to move into the cell. Okay, right, and cause excitation of the postganglionic neuron. Okay, right, so that's the ganglionic type of nicotinic acetylcholine receptor. Now what I want to talk about is some nicotinic acetylcholine receptors that are found in the CNS. And there are two really important ones that are found in the CNS, so I suppose I should call these the CNS types of nicotinic acetylcholine receptor. Okay, so the first one that I want to show you is a Hetero Pentamer, very similar to this Hetero Pentamer that we've seen here, where it's just constructed out of two different types of subunits. Okay, and the second one I'm going to show you is a homopentamer, where it's constructed out of just one type of nicotinic acetylcholine receptor subunit. Okay, so once again, let me firstly show you the Hetero Pentamer. So the Hetero Pentamer is going to be constructed out of two different types of nicotinic acetylcholine receptor subunit, and these two different types are the alpha-4 alpha subunit, okay, of which you're going to have two of those, and then you're going to have two beta subunits, and they're going to be beta-2 type, okay, so beta-2 free. Okay, right, so now let me show this CNS type nicotinic acetylcholine receptor, and basically it's exactly what I've just shown here. Again, the two alpha subunits of the nicotinic acetylcholine receptor are going to be separated by a single beta. Okay, so here are the alpha 4s, like so, and in between these two alpha 4s here you've got a beta 2, and then in these two remaining gaps you're going to have two beta 2s there. Okay, and again, if I colour this in, to show the analogy here, I'll colour the alpha 4 subunits again in green here, okay, and then I'll colour in the beta 2 subunits here. Again, you can see now how similar this CNS type nicotinic acetylcholine receptor pentamer is to that ganglionic type nicotinic acetylcholine receptor. Okay, and the binding sites again are at the positive aspect of the alpha subunits. Okay, so you'll have one here at the positive aspect of that alpha 4 subunit, and then one here at this positive aspect of this alpha 4 subunit. Okay, so this is a nicotinic acetylcholine receptor that is found in the CNS, and again it's going to mediate excitation by acetylcholine of postsynaptic neurons. Okay, so when two acetylcholine molecules bind in those acetylcholine binding pockets, it will cause the channel to open, and again it will conduct sodium ions into the cell. Okay, now what I want to discuss with you is an example of a homopentamer, and this is what's known as the alpha 7 homopentamer, so alpha 7 5. Okay, so let's show this one, and this again is present within the CNS and mediates fast excitatory neurotransmission. Okay, so this basically is just going to consist then of 5 alpha 7 subunits of nicotinic acetylcholine receptors all bound together to make a homopentamer here. So this one's going to be an alpha 7, this is going to be an alpha 7, this is alpha 7, and these two are also alpha 7 subunits. Okay, so I'll colour these all in green as I have done for alpha subunits previously. So these are all identical subunits of nicotinic acetylcholine receptors, and therefore this pentamer is called a homopentamer. Okay, now the same argument that I applied earlier for glucial and ilic and glic applies, which is because of the symmetry of this thing, it has to now have 5 binding sites for acetylcholine. One will be there, one will be there, one will be there, one will be there, one will be there. It can only have 5 or 0. Either the interface works, in which case it works in the case of all of them, or it doesn't work and then it doesn't work in the case of all of them. Okay, so you have to have 5 acetylcholine binding sites for this alpha 7 5 homopentamer. Okay, and again that's found in the CNS. Again, it's a sodium and potassium conductive channel once it opens, and therefore when it opens in response to acetylcholine, it will allow sodium ions into the cell, depolarizing the postsynaptic cell and making the postsynaptic cell more likely to fire an action potential. Okay, so those are some important notable examples of nicotinic acetylcholine receptor pentamers then now. Okay, what I would like to now discuss is a clinical correlate of nicotinic acetylcholine receptors. Okay, so a clinical relation. Okay, and this is the disease Myasthenia gravis. Okay, so firstly, what does myasthenia actually mean? Well, myasthenia is a long word meaning muscle weakness, basically. Okay, so we already have an idea about what this is going to actually entail then, just from the definition of what myasthenia means. Okay, it's going to lead to muscle weakness, which is an inability to generate force in your muscles. Okay. Right, so here is muscle weakness. Right, so let me explain what usually happens in myasthenia gravis. And I should explain right now that there are different causes of myasthenia gravis. However, the major cause, the most common cause of myasthenia gravis is that it's an autoimmune attack, basically. Okay, so it's an autoimmune reaction in which you generate antibody molecules that are targeted against nicotinic acetylcholine receptors, specifically antibody molecules that are targeted against an epitope of the neuromuscular junction type nicotinic acetylcholine receptor. So this one up here, basically. Okay, so let me just explain what an antibody molecule is to make sure that everyone's on the same page. So basically, antibody molecules, or immunoglobulin molecules as they're also called, are molecules produced by the adaptive immune system to attack pathogenic proteins, basically. Okay, now let me show you the structure of an antibody molecule then. So antibody molecules consist of 4 separate proteins which have been joined together. Okay, the 2 proteins that I'm showing you now are the heavy chains of the antibody molecule, and the 2 ones that I'm adding on are now the light chains. So these 4 lines are the 4 different proteins that make up an antibody molecule, and they're connected at multiple points. So the 2 heavy chains here are connected, and each heavy chain is then connected to its light chain. Okay, so these are what are called the heavy immunoglobulin chains. So I abbreviate immunoglobulin down to IG. Okay, and then chain. And this one here is the light immunoglobulin chain. Now, basically antibody molecules, like so, can be divided up into 3 main portions. There are the 2 variable regions of the antibody molecule, which are these portions up here, which are identical to one another, but which differ from other antibody molecules that have been produced by different cells of the adaptive immune system. Okay, so basically the idea is that the adaptive immune system is capable of producing a huge number of different antibody molecules, and all of these different antibody molecules that it's capable of producing will all have different variable regions. Okay, and the variable region is the portion that can bind to other proteins. Particularly, you want it to bind to pathogenic proteins. So when you're infected with a pathogen, you want to produce antibody molecules which are capable of binding to proteins on the surface of that pathogen, basically. Okay, so you want to produce antibody molecules where the variable region can bind to little regions known as epitopes of the proteins on the surface of the pathogen. Okay, so the variable region is also therefore called the FAB region, which stands for fragment antigen binding. Okay, so there is this key word which is antigen, and antigen is just any protein, or molecule rather, more broadly than protein, that can have an antibody produced against it, basically. Okay, so if we imagine that our antigen is a protein, then I might represent it like so. Basically, there are loads of different facets of the antigen molecule which could potentially have antibody molecules made against them, and little facets of the antigen molecule, like for instance this little corner here, are known as epitopes. Okay, so basically the adaptive immune system is capable of producing a huge number of different antibody molecules where they have different variable regions, and you can produce antibody molecules with variable regions that can bind to different epitopes of different antigens, basically, and this is an important part of the adaptive immune system. Okay, then there is this other portion of the antibody molecule, which I'm now highlighting in orange, and that's the constant region. Okay, that's the portion that doesn't vary between different antibody molecules. Okay, so this is called the constant region, and this doesn't participate in the binding to the antigen epitope, basically. Okay, so to summarize then, the adaptive immune system can produce a huge number of these antibody molecules where they all have different variable regions, okay? And when it gets exposed to some antigen from a pathogen, it can produce loads of different antibody molecules which have variable regions which can bind to epitopes of this antigen, basically. And these antibody molecules will then coat the pathogen, is the idea, and the constant region of the antibody molecules can then recruit other proteins or other cells potentially, which can then attack the pathogen, basically. It can recruit other arms of the immune system, basically, to attack the pathogen. Right, so what happens in Myasthenia gravis is you end up producing antibody molecules with variable regions which recognize some epitope of the neuromuscular junction nicotinic acetylcholine receptor. Okay, so they bind to neuromuscular junction nicotinic acetylcholine receptor. Okay, and what this overall leads to is a humongous great reduction in the number of nicotinic acetylcholine receptors on the surface of the skeletal myocytes. So to cut a long story short, okay, obviously you get the antibody molecules binding to the neuromuscular junction nicotinic acetylcholine receptor. That can then lead to the adaptive immune system, it can then lead to the immune system then attacking the portions of the membrane with those neuromuscular junction nicotinic acetylcholine receptors in them. And to cut a long story short, what it then overall leads to is the levels of the nicotinic acetylcholine receptor in the skeletal myocyte going down, basically. So you end up with far fewer nicotinic acetylcholine receptors in your skeletal myocytes. Okay, now what then is the effect of that? Well, that means that the skeletal myocytes now are effectively insensitive to acetylcholine, because remember, to respond to acetylcholine, they need to have the nicotinic acetylcholine receptors on their surface. If you've taken those away, they're not going to respond nearly as well to the acetylcholine. And that's why this produces muscle weakness, basically. Okay, because now you can make your neurons fire as much as you like. They can release loads of acetylcholine onto the skeletal myocytes, but you're going to struggle to make your skeletal myocytes actually contract, okay, because the skeletal myocytes have lost their nicotinic acetylcholine receptors. Okay, right, so then what are the key symptoms of Myasthenia gravis? Okay, well, key symptoms are that you can end up with muscle weakness all over the body, so you'll have difficulty in generating force in your leg muscles and your arm muscles. You'll end up dropping heavy objects, things like that. Okay, you also often end up with something called diplopia, okay, which is basically a fancy word to mean double vision. Okay, so this is when you see an object twice, basically. Okay, and you can make yourself see an object twice, okay, by trying to get your eyes to not both focus on the object, basically. So if you try to force your eyes to look in opposite directions, you can end up seeing an object twice, basically. Okay, and they'll be separated in space. So that's what double vision means, seeing an object twice, because your eyes aren't both focused in the right way on the object, and that's occurring because of problems in the control of the extraocular muscles. Okay, so because the extraocular muscles aren't responding in the way that they should to acetylcholine stimulation, you're now not getting your eyes in the right positions, okay, and that leads to seeing double, okay, and the fancy word for that is diplopia. Okay, in addition, you often end up with ptosis, okay, which means drooping of the eyelids, okay, and again that's because the muscles which keep the eyelid up are becoming weak, so the eyelids end up drooping down, okay. Other more very serious things are that you'll have problems with chewing, problems with talking, okay, so chewing and talking can be problematic. And the way that it can lead to death is if the diaphragm becomes too weak, basically, because of course the diaphragm, it's a skeletal muscle, basically. It's controlled by neurons which are releasing acetylcholine onto it. Okay, so if the diaphragm becomes too irresponsive to the acetylcholine and becomes therefore too weak, it's too difficult to actually get it to contract, then of course what you can end up with is respiratory failure. Okay, so it can lead to respiratory failure, okay, which may require mechanical ventilation. Okay, so respiratory failure. Right, okay, so that's Myasthenia gravis and the nicotinic acetylcholine receptors. Okay, let's now move on to another type of Cys-Loop ligandated ion channel family present in humans, okay, which is the Gaba A receptors. Okay, so next up, let's talk about the Gaba A receptors. So firstly, just like we did with the nicotinic acetylcholine receptors, I want to now talk about the fact that this is a massive, great family of receptors. It's not just one receptor. Okay, so again, the beautifully simple thing would be if there was just one subunit for a GABA A receptor, which we could call the GABA A receptor subunit, and all we had to do to create a GABA A receptor would be then to join together 5 GABA A receptor subunits, and we'd have our GABA A receptor. Okay, but it is not that simple, unfortunately. In reality, there are multiple different GABA A receptor subunits in humans, just like there are multiple different nicotinic acetylcholine receptor subunits. Okay, in fact for GABA A receptors it's even more than there are for nicotinic acetylcholine receptors. Okay, so there are 19 different subunits in humans which are all subunits for GABA A receptor pentamers. Okay, right, and these are grouped into many, many different families. Okay, so let me give you the names of the different GABA A receptor subunits then. So firstly, there are 6 GABA A receptor subunits which are counted as alpha subunits. Okay, so there is the GABA A receptor alpha 1 subunit, the GABA A receptor alpha 2 subunit, etc., all the way on to the GABA A receptor subunit alpha 6. Okay, now I want to stress these are completely different from the alpha subunits of nicotinic acetylcholine receptors. They're not the same proteins. Okay, they are different proteins. Of course, they are both Cys-Loop ligandated ion channel subunits. So they do have those common structural features that we've discussed, but they're going to participate in very different types of receptors. These subunits are going to help build GABA A receptors, receptors for GABA, which are chloride channels. Okay, whereas the alpha subunits of nicotinic acetylcholine receptors are obviously going to build receptors for acetylcholine, which are sodium and potassium ion channels. Okay, right, so you have 6 alpha subunits, then you have 3 beta subunits, which are called beta 1 to beta 3, okay. You also have 3 gamma subunits, which are called gamma 1 to gamma 3. Oops, I've done it wrong, but never mind, I'll just sort of put gamma 1, gamma 2, gamma 3, like so. Okay, then after those you have the delta and epsilon GABA A receptor subunit, and again I'll stress these are separate from the delta and epsilon subunits of nicotinic acetylcholine receptors. Then you have another family of GABA A receptor subunits, which is the row family, okay, which is row 1 all the way through to row 3. Okay, and let's see where we are at the moment. So we've got 6 here, we've got another 6 amongst those two, another 3 here, that takes up to 15, 16, 17, so we only need 2 more now, and the final 2 are called pi and theta. Okay, and that takes us up to the 19 different GABA A receptor subunits. Okay, right, so just as we had with the nicotinic acetylcholine receptor subunits, we have a huge number of different GABA A receptor subunits, okay, and you could combine these together in so many different ways, and therefore there are a huge number of different GABA A receptor pentamers that are possible, basically. However, not all of the possible combinations actually are found in humans. Okay, so what I'm now going to tell you about is the major types of GABA A receptors that are actually found within the central nervous system of humans. Okay, now the major type of GABA A receptors only include alpha, beta, and gamma GABA A receptor subunits. So forget about these lower ones for now. Okay, it's the alpha, beta, and the gamma that form most GABA A receptor subunits. Well, most GABA A receptor pentamers rather. Okay, so the type of GABA A receptor pentamer then that I'm going to show you is going to be constructed from alpha, beta, and gamma receptor subunits. Okay, and let me show you how they're going to be arranged relative to one another. Okay, so again we're going to be looking down at the pentamer from the extracellular aspect. Okay, so here is the pore down the middle, and again you're going to have 5 separate subunits making up the full GABA A receptor here. Okay, now basically in these alpha, beta, gamma type GABA A receptors, you're going to have 2 alpha GABA A receptor subunits, 2 beta GABA A receptor subunits, and 1 gamma GABA A receptor subunit. And let me now show you how they're going to be arranged. So basically they're going to be arranged in clockwise order: alpha, beta, and then gamma. Okay, so let me just colour these in. Okay, so we'll have the alpha subunits here coloured in blue, okay, like so. Then we'll have the beta subunits coloured in green, okay, and the gamma subunits, we'll have those in red, I think. Okay, right, so most GABA A receptors then, most of the GABA A receptor pentamers consist of 2 alpha type GABA A receptor subunits, 2 beta type GABA A receptor subunits, and then 1 gamma type GABA A receptor subunit, arranged like so. Okay, so let me give you some examples of alpha, beta, gamma GABA A receptor types, okay, where I give you this specific alpha subunit that is used, the specific beta subunit that is used, and the specific gamma subunit that is used. And I should just stress right now that when you're building one of these alpha, beta, gamma GABA A receptors, the alpha subunits that you're going to put here and here are going to be the same alpha subunits. So you're going to pick one of the alpha subunits of GABA A receptors to put in these 2 sockets. You're going to pick 1 beta subunit of GABA A receptor subunits to put in these 2 sockets, and you're going to put 1 type of gamma receptor subunit there. Okay, right. So, in fact, before I tell you examples of alpha, beta, gamma GABA A receptors, let me tell you about where the ligand binding sites for GABA actually are. Okay, so the binding sites for GABA are between the alpha and the beta subunits. Okay, so here at the negative aspect of the alpha subunit here and the positive aspect of the beta subunit, that is a GABA binding site. Okay, similarly over here at this negative aspect of this alpha subunit and the positive aspect of this beta subunit, that's another GABA binding site. So here are the 2 binding sites for GABA basically. Okay, right, so each GABA A receptor of this alpha, beta, gamma type, where you've made them in this way, has 2 GABA binding sites. Okay, and when GABA binds to those two GABA binding sites, then it will be induced to open, and it will open a chloride channel, which will then allow in inhibitory currents into the postsynaptic neuron. Okay, so there is one combination then of alpha, beta, and gamma subunits that is found most commonly within the brain, and is the one that, if you're going to memorize any of them, is the one that should be memorized. Okay, so this is alpha 1, 2. Okay, so in the alpha sockets here, you put the alpha 1 subunits. You put an alpha 1 here and an alpha 1 here. Okay, then you have beta 2, 2. Okay, so in the beta sockets here, you put the beta 2 subunit, and then finally the gamma subunit will be gamma 2. Okay, and you might like to put brackets around that as well. Okay, so this is the main type of GABA A receptor that is found within the brain. It's one of these alpha, beta, gamma type GABA A receptors, where the alpha subunit that you use is the alpha 1 subunit, the beta subunit that you use is the beta 2 subunit, and the gamma subunit that you use is the gamma 2 subunit. Okay, so that is the most common GABA A receptor found in the human brain. Okay. Another notable example of one of these alpha, beta, gamma GABA A receptors, okay, consists of this makeup. Okay, so the alpha subunit you use is alpha 2. So in both of those sockets, you'll put an alpha 2 alpha subunit of GABA A receptor. The beta subunit that you use is beta 3. Okay, so in both of these sockets, you'll put the beta 3 subunit. Okay, 2. And then the gamma subunit that you use is gamma 2. Now, often the gamma subunit that turns up in these alpha, beta, gamma, GABA A receptors is the gamma 2 subunit. Okay, it seems to be the main gamma subunit that's used, and it seems as though it has roles in actually targeting the GABA A receptor to the postsynaptic membrane where it's involved in a synapse. Okay, so it seems as though gamma 2 is really, really important in getting the GABA A receptor targeted to a postsynaptic membrane where it can actually be involved in a synapse. So most of the main GABA A receptor pentamers, which are alpha, beta, gamma type GABA A receptors, do have the gamma 2 subunit in. Okay, the final one that I just want to note is alpha 3 is the alpha subunit that you use. So in these two sockets, you'll put the alpha 3 subunit in. Okay, the beta subunit that you use is beta 3, so you'll put in 2 beta 3s again, and then the gamma subunit is gamma 2. Okay, so very similar to this one here, except that we've swapped the alpha subunit for alpha 3 in place of alpha 2. Okay, right. So those are 3 really important examples then of GABA A receptor pentamers, which are all these alpha, beta, gamma type GABA A receptor pentamers, okay, and which are found in the brain on postsynaptic neurons, and which will respond to GABA by opening a channel which will allow chloride anions into the cytoplasm of the postsynaptic cell, and therefore will hyperpolarize the electrical potential difference across that postsynaptic cell membrane. Okay, right. We'll call it there for this video, and then in the next video what we'll discuss is the benzodiazepines, which are a clinical correlate of GABA A receptors, and we'll also discuss a form of heritable epilepsy, which is caused by mutations in GABA A receptor subunits.